Good afternoon, everybody, to the fourth lecture in our autumn series, this being the 10th series that the Guy Foundation has done. The series is on the theme of genes and metabolism, bioelectricity, and the quantum spark of life. Our speaker today is Nick Lane, Professor Nick Lane. We're really pleased, Nick, to have you speaking to us again today. The title of Nick's talk today is Proton-motive CO2 fixation, chirality, protometabolism, and the emergence of genes. And without further ado, I'm going to hand over to Rhys. So Rhys Mould is our research fellow at the Research Centre for Optimal Health at the University of Westminster. And Rhys, you're going to introduce Nick this afternoon. So over to you.
All right, good morning and afternoon, everyone. My name is Rhys Mould, and it's my pleasure to again introduce Professor Nick Lane to the Guy Foundation seminar series. So you must forgive me if I've reused some slides. So Professor Nick Lane is Professor of Evolutionary Biology at the Centre for Life's Origins and Evolution at UCL. And today's talk, entitled Proton-motive CO2 fixation, chirality, protometabolism, and emergent genes. So Professor Lane is incredibly well established with over 100 peer-reviewed papers, and notably also as an author with 5 acclaimed books selling over 150,000 copies worldwide, including Transformer, The Vital Question, Life Ascending, Power, Sex, Suicide, and Oxygen. And with numerous achievements and accolades, including founding member of the UCL Consortium for Mitochondrial Research, the 2015 Biochemical Society Award for Outstanding Contribution to Molecular Life Science, and the 2016 Royal Society Faraday Prize and Lecture of the UK's Premier Award for Excellence in Communicating Science. Professor Lane's research focuses on 3 major transitions in evolution: the origin of life, the origin of the eukaryotic cell, and the evolution of conserved traits. Today, Nick is going to talk to us about the first point, the enigmatic origin of life. So there's a few theories about the origins of life. Prominently, I'm going to talk about 2 real quickly to set up the scene for today's talk. So the first, the primordial / prebiotic soup, the RNA world hypothesis. In here, organic molecules began from cyanide derivatives catalyzed by UV radiation. Interestingly, theories about where the cyanide first come from includes meteorite impact, which I think is pretty cool. So nucleotides in this soup eventually polymerized to RNA. But then what? There's some problems with the theory, and the transition from the RNA to the system that resembles metabolism is particularly problematic in this theory. So we move on to the second, the hydrothermal vent or metabolism first hypothesis. So hydrothermal vents are these rocky chimneys on the surface of the ocean that billow hot water and minerals into the ocean. So you have this hydrogen-rich water from deep within the earth meeting the carbon dioxide-rich ocean water. This feeds into a reaction system that resembles what would become modern metabolism. The problem is that hydrogen and carbon dioxide are nominally unreactive. So the question is, how do you get these precursors to metabolism into what would resemble genes today? Perhaps the answer ties nicely into the theme of the autumn series of electricity, the flow of charged particles. So do vents with alkaline interiors and acidic interiors resemble cells and use a proton-motive force to drive carbon dioxide fixation? This diagram taken from a review authored by Nick. So you start with the geochemical carbon dioxide fixation at the bottom of the diagram, and you can see working your way up from protocells to protometabolism into eventually eukaryotic archaea and bacteria. So without much further ado, I'm going to hand straight over to Nick. And for the title of his talk, as I say again, proton-motive carbon dioxide fixation, chirality, protometabolism, and the emergence of genes. So what promises to be a really fascinating talk.
Thank you very much for that very kind introduction. Okay, I'm not going to go over that title again. So as Rhys pointed out, there's a long history of kind of controversy and anger and just generally disagreement about the origin of life. It's one of the most interesting and difficult questions in science. And it's going to be basically impossible to prove it or disprove it one way or the other. And so people can spend their entire careers working on this question. And I suppose if you spend 30 or 40 years working on something, then the last thing you really need is for some upstart to come along and disprove everything you or spent your life doing. So there tends to be, people tend to get into rather entrenched positions and actually rather sadly stop talking to each other after a while. So for decades, really, the origin of life was seen as a question in chemistry, and biologists kept well away from it. Biologists mostly regarded the origin of life as I suppose, armchair speculation of the worst kind. And chemists saw it as a very much specific question in synthetic chemistry. Can we make nucleotides starting from whatever? And will that give some clues into how life might have started? And they've done a very good job of it, but they've started from compounds like cyanide, and there are these questions about not only where does the cyanide come from, but also how would then an RNA world, as Rhys said, invent metabolism. There's some seriously difficult questions there. Biologists have entered really, since the advent of really serious phylogenetic sequencing of the tree of life, going all the way back to the most ancient bacteria and archaea. And so now we have a reasonable indication of the overall topology of the tree of life and what the earliest cells might have looked like. So we don't have much agreement even among biologists about that. But if you take a step back from this kind of very fine-grained gene sequencing to some overall kind of concepts, what I'm going to put to you is that life is in fact an excellent guide to its own origin, although that sounds circular, in three specific aspects: in energy, in the way that cells drive their chemistry through chemiosmotic coupling and proton-motive force, so effectively electrically charged membranes. Metabolism, which is universally conserved at the level of chemistry, and genetic information. We're all, of course, familiar with the universal genetic code, which has patterns which seem to say something about the origin of life as well. So just a quick outline of what I mean by these three things. So energy, I would hope that you'd all be familiar with this. So this could be a bacterial cell. Let me just put this to a pointer. Or it could be a mitochondrion in our own cells, effectively breaking down food, glucose in this case, to acetyl-CoA into the Krebs cycle. And the Krebs cycle at its simplest is pulling out CO2, which we excrete, and hydrogen in the form of hydrogen atoms, in the form of NADH in our own case, but hydrogen atoms which are split into the protons and the electrons. So we have a current of electrons, this yellow dotted line here, to oxygen. And that current of electrons powers the extrusion of protons across the membrane, giving the proton-motive force, which in this case powers ATP synthesis, which is a kind of an electrical turbine, which I will come on to later on. So this charge across the membrane, it's around about 150 to 200 millivolts. But if you were to shrink yourself down to the size of a molecule and stand next to that membrane, then the field strength you would experience is about 30 million volts per meter, which is equivalent to a bolt of lightning. So it's a pretty intense electrical charge on that membrane. So that's roughly how aerobic life works. You could substitute oxygen for alternative electron acceptors, nitrate, for example, nitrite, these kind of things. And the hydrogen doesn't have to come from food. It could come from gases like hydrogen sulfide or even metals like ferrous iron. So this is an overall concept as to how vectorial energy generation works in cells. So that's energy generation. This probably looks the same. It's intended to look the same. It's slightly different, and in fact, it's exactly the opposite. So here we have the Krebs cycle again, but the arrows are reversed. Now we're pulling in hydrogen, we're pulling in CO2, and the electrical membrane potential is driving their reaction to effectively drive the reaction of gases, hydrogen and CO2, to form Krebs cycle intermediates, so the carboxylic acids that are right at the heart of metabolism. And from these carboxylic acids, bacteria, archaea, and indeed we ourselves generate amino acids, sugars, nucleotides, lipids, and so on. So all of the basic building blocks of life are derived from the reverse Krebs cycle. It's slightly longer than the Krebs cycle, the oxidative Krebs cycle that most people are more familiar with, and it includes things like pyruvate, for example. So it's things that kind of mammalian biologists would say, no, that's cytosolic. They are, in fact, in the reverse Krebs cycle. So it really is the heart of metabolism as we know it. So we have this tension between these two processes, one of which is more or less burning carboxylic acids, burning hydrogen to produce energy in the form of ATP, and the other one which is using that energy to drive the reaction of gases to form the organic precursors of life. So the third aspect then, information, the genetic code. There are patterns in the genetic code, and I'll come back to this at the end of the talk, which suggests that there were direct physical interactions, stereochemical interactions between amino acids and the bases in the genetic code that code for them. Now, I'm showing two possibilities of the way that these might work. It could simply, these little colored lozenge things here are the amino acids, and the bases are of different sizes. It could be that we have a tRNA-like pocket that amino acids are binding into, or it could be that they're somehow just binding to a messenger RNA type. We don't know, but there are definitely physical interactions are suggested by the code. And the important point about that is if you have a random sequence of RNA and there are non-random interactions with amino acids, which if they polymerize there would mean any random RNA would give you a non-random peptide. The properties of the peptide would depend on the sequence of the RNA, and that can be random, but it would give you a non-random peptide. It would give you a peptide that depended on the exact sequence or the properties of the RNA. And that's a very simple mechanism for bringing information into biology. So I'm going to come back to that right at the end, but I just want to set that up to begin with. So a general point that emerges from this, which is actually in a way quite thrilling, in another sense quite scary, is that what this means is that a geochemical, at the origin of life, a geochemical proton-motive force is driving directly CO2 fixation and a protometabolism before we have any genes or enzymes, and that itself gives rise to genes through direct physical interactions. And that says all this has to happen in the same place at the same time. So we can't have the planet as a laboratory. We can't have some chemistry happening in the clouds and some chemistry happening in the hydrothermal vents and some in terrestrial geothermal systems, and then somehow these things come together over what, years, decades, hundreds of years, millions of years. There's all kinds of claims out there. This is altogether more bracing. It says this is effectively synthetic chemistry happening in one place at one time. And in principle, that means it can be done on the benchtop in the lab. That's a pretty daunting prospect for a PhD student, but nonetheless, it's an experimentally tractable question. So are there any places imaginable? Well, there are. So the best prospect, I think, was discovered by Deb Kelly nearly 25 years ago now, which is Lost City, which is about 15 miles off the axis of the mid-Atlantic ridge. And it's not actually, this is not quite like the hydrothermal vents that Rhys showed at the beginning. Those were black smoke events. These are not smoking, notice, but they are in fact actively venting. They're a different kind of vent produced by the reaction between effectively minerals like olivine, and a lot of the mantle of the earth is made up of olivine, and water. So water will percolate down into the crust, and if there's olivine relatively close to the crust, and there is at the spreading centers of the mid-ocean ridges these days, then you get this reaction between rock and water, which is effectively oxidizing the iron, the iron in olivine, and producing hydrogen gas in alkaline solution. Alkaline meaning pH 11 or thereabouts. So these vents were discovered by Deb Kelly in the year 2000. She's holding a lump of a black smoker vent in her hand here with the chimney in the middle through which the smoke would belch out. And then in her right hand, she's holding a piece of lost city, and it looks a little bit like a mineralized sponge. So it's really an interconnected labyrinth of micropores, which have vaguely cell-like structures. So this idea that alkaline hydrothermal vents could be electrochemical flow reactors was put forward by Mike Russell in really as early as 1989 and developed through the 90s, and then really compellingly with Bill Martin from 2003. Several papers, another one, these two papers I'm citing here are absolute classics that have driven people into the field working on the origin of life. A whole generation now of people with background in biochemistry and microbiology who are recognizing the origin of life as a question that biochemists and microbiologists may have something to say about rather than only chemists. I shall touch on physics in a little while. So what you're seeing in this figure from that 2003 paper is a lot of chemistry happening inside these micropores, eventually ending up with a divergence of bacteria and archaea with different membrane types, and but with ribosomes, with DNA, with RNA, and so on. So the common ancestor was not a free-living organism, according to this theory, but was effectively part of the vents, almost built out of the pores in the rocks themselves. So it's a radical idea, and it certainly, I found it really quite difficult to grapple with when I first heard it put forward 20-something years ago. Now I think they're along the right lines, but there's lots of, again, arguments between the different protagonists here, and this is my own take on it. So Bill Martin and Mike Russell really don't agree with each other about all of this anymore, and I'm not sure that either of them would necessarily agree with what I'm telling you now, but this is the way that my group sees it now. So imagine this is one of the pores in the vents. We have acidic ocean waters percolating in. We have alkaline fluids coming from the hydrothermal fluids with hydrogen gas in it, CO2 in the oceans, and a mineral barrier around here. These vents are really iron sulfide minerals are included in these barriers, or at least we think they would have been 4 billion years ago when there was no oxygen in the ocean. So the ocean chemistry was different. In the absence of oxygen, there were more transition metals there, nickel, iron, molybdenum perhaps, and these would have precipitated into these barriers. So this is roughly what I showed you. We have a reverse Krebs cycle here with hydrogen and CO2. The proton-motive force from the acidic oceans and the alkaline hydrothermal fluids is driving the reaction, catalyzed by iron-sulfur minerals of hydrogen and CO2 to form the most thermodynamically favored products, which are amino acids and lipids. So this is theoretical thermodynamics from people like Tom McCollum and Jan Amond. And so theoretically, these should form spontaneously under those conditions. And I'll show you that there's some truth in that. So if we can form lipids, then perhaps they were precipitated as a bilayer membrane inside this, almost templated by this cell-like pore. And this little crystal here would then be a nanocrystal, a nanoparticle of some sort of an iron sulfide associated with the membrane now, and so still driving CO2 fixation with the same overall topology, with protons crossing this barrier and entering, and CO2 fixation happening inside now a partially open, partially closed system. These fatty acid bilayers are pretty leaky to protons, to hydroxide ions, and so on, but they would tend to retain higher concentrations of things like amino acids, and so you could possibly drive that chemistry a little bit further, even as far as nucleotides. And then in the final section here, I'm showing our view of an RNA world, where we're beginning to see interactions between RNA and amino acids, but as part of a much richer chemistry involving this whole protometabolism. So this is a hypothesis, and it is at least testable. And we've spent the last 10 years, my lab, really trying to test as many aspects of this as we can, and some of it's worked quite well, and some of it's taken an awful long time. But I want to tell you about it because we are pretty excited that it feels coherent, and there's at least some evidence backing this view. And it will also touch. There's not much quantum in here at the moment, but I will touch on some quantum biology in relation to this. So I'd like to go through these three themes of energy and metabolism and information, and I'm going to insert chirality in the middle of it because it's something that I became interested in quite recently, and I'm not sure it's gone in the direction I would like it to have gone, but I'll just give you a flavor of that as well. So life is powered by the proton-motive force. This idea goes back to Peter Mitchell. In 1961 was when he put forward the chemiosmotic hypothesis. This picture is with Geoffrey Mould in, I think, 1946. So they were together in Cambridge, and they worked together throughout throughout their lives. It was a very close working partnership. And Geoffrey Mould, I don't think, has got the credit that she deserved, in my opinion. I never met either of them, so I don't really know. But she was the experimentalist. She was a brilliant experimentalist. And in writing my last book, I reread all of their papers from the 1960s. And I have to say, the ones where Geoffrey Mould was an author, I found them much clearer and much more modern in their terminology. So I think she made a bigger difference to the hypothesis that's usually credited to Mitchell than is generally recognized. But the overall idea certainly does go back to Mitchell, this idea of chemiosmotic coupling, and it's genuinely counterintuitive. Nobody imagined that was how bioenergetics was going to work. There's a lovely quote from Leslie Orgel, who himself was a pioneer on the origin of life, who said, "Not since Darwin has biology come up with an idea as counterintuitive as those of, say, Einstein, Heisenberg, or Schrödinger," ironically, the pioneers of quantum mechanics. So Mitchell also had been to a conference in Moscow in 1957 on the origin of life. So this was before his chemiosmotic hypothesis. And there's a lovely quote from that meeting, from his talk there. He said, "I cannot consider the organism without its environment. From a formal point of view, the two may be regarded as equivalent phases between which dynamic contact is maintained by the membranes that separate and link them." So when he's saying organism, think bacterial cell, don't think human being or something. And he's saying that the outside world, the environment and the inside of a bacterial cell are equivalent phases, which is not the usual way of thinking about these things, separated and linked by a membrane 5 nanometers thick. So it's really quite an unusual statement. But if you think about the origin of life before there is such a thing as a cell, an organism, and the distinction between what becomes a cell and the environment is certainly less clear cut, then this idea effectively of a phase difference across a membrane across which ions and so on will move is a very, I think, productive way of seeing the origin of life. So if we put this in terms of hydrothermal vents, here's this pore again in an alkaline hydrothermal vent. We have alkaline fluids inside, we have acidic ocean waters coming to the outside, and we have a barrier around it. Now, this is basically the same in terms of the topology as a bacterial cell, which is pumping protons out, as Mitchell recognized. So it's relatively alkaline inside and relatively acidic outside. Now, there are differences in terms of the electricity here, which is to say, if you pump a positive charge outside, then you're moving the charge outside, and so you're generating electrical potential across the membrane. That's not the case here. These are electrically balanced solutions on both sides of this. So you don't have a charge here, you have a difference in proton concentration. And if a proton moves in faster than anything moves out, then actually you end up with the reversal of this topology with a net positive charge inside, which will be balanced by the movement of other counterions. So it's not as exact as I would wish it would be, but nonetheless, there's an equivalence, broad general equivalence in the topology here. And the question then is, can we go from one to the other? And I've already shown you the hypothetical idea as how you might do that transition. So just to put a bit of meat on the bone with this, so this on the left-hand side is my kind of simplistic depiction of a methanogen. So methanogens are growing from CO2 and hydrogen over here. Methanogenesis as a process is actually the production of methane. Methane is a waste product of methanogenesis, and it's just lost to the environment. And so the only thing that's retained, that's conserved by the process of methanogenesis, is an ion gradient across a membrane. And I am asserting here that it's a proton gradient. It's often a sodium gradient, but that sodium gradient is converted into a proton gradient as well. So what I'm showing is this protein in the membrane called the energy converting hydrogenase, which is proton motive, which is to say protons move through from this side of the membrane to the other side of the membrane. And that proton motive force drives the reaction between hydrogen and oxidized ferredoxin to reduce the ferredoxin. And the reduced ferredoxin then reduces CO2 in the end to a methyl group bound to a cofactor. The R is a cofactor, and on into central carbon metabolism. There's ATP requirements in central carbon metabolism as well, but it's really this little bit over here, the protons, the hydrogen, and the ferredoxin, and this membrane protein that are critical. Now, on the right-hand side, here's a schematic of the ECH, and there are several iron-sulfur clusters in here, 4Fe, 4S clusters. There's 3 of those, and there's 1 nickel-iron cluster. And this is the energetics I'm showing here is taken from Rolf Tauer, from one of his papers on methanogens, and it shows really what the issue is. So the reduction potential of hydrogen in the kind of environment that most methanogens live in is around about minus 300 millivolts. So that's assuming a partial pressure of 10 pascals of hydrogen. So minus 300 millivolts. Whereas ferredoxin, when it's 99% reduced, has a reduction potential of about minus 500 millivolts. So hydrogen with minus 300 millivolts is reducing ferredoxin minus 500 millivolts, which shouldn't happen. It does happen because of the proton motive force. Now, the easiest way for me to understand that is the binding of protons. So this is the proton pore in the membrane. Protons are binding close to these iron-sulfur clusters, and these two of these iron-sulfur clusters have been shown to be pH sensitive in their redox potential. I'll come back to that in a moment. But what it means is if protons are bound, then they become they become less reducing or more easily reduced. In other words, hydrogen could reduce them. They will probably have then presumably a redox potential in the range of minus 300 millivolts and can be reduced by hydrogen. But then if those protons detach, then the redox potential will immediately drop by a couple of 100 millivolts. So it now becomes much more reducing. You could no longer reduce it with hydrogen, but this system is now capable of reducing ferredoxin. So it's dropped down to close to minus 500 millivolts. So this is a very simple proton-powered redox switch, which allows hydrogen, which is not sufficiently reducing, to reduce ferredoxin that can reduce CO2. So this is hypothetical, but there is some evidence backing that up. I'm not going to go into that in detail today, but we can talk about it afterwards if you have any specific questions. So these iron-sulfur clusters, they're in ferredoxin, they're in the energy-converting hydrogenase, and they can be formed, as I'll show you, they can be formed fairly spontaneously under prebiotic type conditions, and they share a very similar half-cell structure with minerals like greigite. So this is again, the 4Fe, 4S cluster is the half-cell structure of greigite, the kind of minerals that you may have seen in the walls of these vents 4 billion years ago. So there's a continuity here, which I find very pleasing, and a way effectively of downsizing a mineral to simple clusters that can do the same chemistry, which are embedded within proteins. So it's not a whole membrane; it's just this kind of fragment of a mineral inside that protein. So just to clarify why hydrogen won't reduce CO2, the redox potential of hydrogen, this is reduction potential here, becoming more negative as we go up, in other words, more reducing as we go up. If you're not fond of electrochemistry, all you really need to appreciate is that hydrogen is not going to reduce CO2 to formaldehyde, it's uphill. It's you're going from the minus 300 or thereabouts to the minus 500, slightly different numbers here. That's if it's at pH 7, but if we're at pH in a vent, we have hydrogen is in alkaline solution at pH 11 and CO2 in acidic, mildly acidic solution at pH 6. And because of the Nernst equation, the redox potential shifts by about 60 millivolts per pH unit. And so hydrogen becomes more reducing at alkaline pH, CO2 becomes more easily reduced. And so that now it's a downhill reaction. And it's very easy kind of intuitively to appreciate that if if hydrogen's in an alkaline solution, it passes on the electrons to something else. What's left behind are protons, and the protons will immediately react with hydroxide ions. It's just a neutralization reaction, and so that will be favored and will be basically instantaneous. CO2, if it picks up an electron, acquires a negative charge, it's not going to readily pick up another electron unless that charge is balanced. If it's in a mildly acidic environment, it picks up a proton very easily. Now it can pick up another electron, another proton, and so on. So it's easier to reduce CO2 in a mildly acidic environment. For that to happen, you would need two phases, one with hydrogen in an alkaline phase, one with CO2 in a mildly acidic phase, and some kind of semiconducting barrier between the two phases. And so we're straight back to Mitchell's conception. And this is something which we can set up experimentally. I was going to say quite easily, I mean, conceptually quite easily. Turns out to be much more problematic than I would ever have imagined. But this is a microfluidic chip. This is the setup inside the hood over here. This work's being done by Fei-Shu Liu and Hanadi Ramu. So these are gas pumps. And we have two channels, one with alkaline fluids containing hydrogen gas at pH 11, sodium sulfide, and sodium silicate. And then the ocean waters, the other channel is CO2 gas around about pH 5, and with ferrous chloride, so iron 2 chloride, and nickel 2 chloride at about a fifth of that concentration. So you spontaneously precipitate immediately a barrier down the two channels. We have more or less laminar flow as they come in and come down these channels, and you precipitate this barrier between them. And the question is, now we have two phases separated by a semiconducting barrier. Can we reduce CO2 with hydrogen that way? And the answer is yes, sometimes, not every time. And sometimes we get contamination, sometimes from the gas cylinders, which can be quite infuriating. But here's a nice clean example. We've done all kinds of controls. This particular control I'm showing you here is with, in fact, instead of a freshly precipitated barrier, it has a copper barrier chip. But what we're seeing in this freshly precipitated iron nickel sulfide barrier, this is NMR, we see a good peak for formate. We see methanol, with some uncertainty still about methanol. Acetate, we're certain about acetate. Pyruvate, some uncertainty about that. Most of these have also been shown with either HPLC or GCMS as well. But we've yet to publish this because we haven't really got a succession of clean runs that we're really pleased that, okay, we've definitely nailed it. A couple of things we do generally find is if you don't have any nickel in that barrier, it doesn't work. If you don't have a pH gradient of more than about 4 or 5 pH units, it doesn't work either. So these are kind of just general observations, but until we've really nailed all the controls, then I better not say any more about that. So just to reorient you to where we are, we've kind of done this first section. We've been reducing CO2 to get to the beginning of Krebs cycle intermediates, and bear in mind, acetate, pyruvate are part of the reverse Krebs cycle. So can we get a little bit further? Can we, if we could make fatty acids, for example, would they precipitate as a bilayer membrane? And would we be able to drive CO2 fixation with some kind of smaller iron sulfur nanoparticle that's associated with this membrane? So would we be able to drive the following chemistry inside? And the literature says no way. So these vents, these alkaline hydrothermal systems, they have pH ranges from about 5 in the early oceans to 12 in some systems. Temperature usually between about 50 and 100 degrees centigrade. They tend not to get much above 100. So these are, geologically speaking, warm rather than hot. Ocean salinity, 600 millimolar practically of sodium chloride. And then divalent cations, which can play havoc with bilayer membranes. So magnesium, calcium in the oceans. So these are the challenges. Can we make under hydrothermal conditions protocell-like things? Well, there has been some work done. In fact, we are trying at the moment to synthesize fatty acids following the biochemical pathways. But 20 years ago, more than 25 years ago, there was a paper from Tom McCollum doing a form of what's called Fischer-Tropsch synthesis, Fischer-Tropsch type synthesis, which is, they were actually using formic acid at quite high temperatures to break it down into hydrogen and CO2, and then the hydrogen and CO2 would react to form long chain hydrocarbons. So these numbers here are the carbon chain length. So 14, 15, 16, this is 16 carbons in a chain of a hydrocarbon chain. And over here, these are alkanols and alkanoic acids, so fatty alcohols and fatty acids, 7, 8, 9, 10, 11. So slightly shorter chains, but nonetheless, these are fatty acids, the kind of things that form membranes, and fatty alcohols. So instead of a carboxylate group, you just have an OH group at the end of the hydrocarbon chain. So starting with them, we did some experiments. So the literature says you can't form these things, but the literature mostly looks at single fatty acids. So decanoic acid, for example. This is optical density, so the light scattering. What we see, or what everyone sees, is at higher pHs, you get micelles, which don't have a lumen. They're just tiny little things. They don't scatter the light much, so very low optical density. If we have acidic pH, you have much higher optical density. These are lipid droplets. Everything gets protonated. You have lipid droplets. They scatter the light a lot. And this region in the middle is where you would expect to see vesicles. So really only at pH 7. But if we have mixtures of 6 different fatty acids, then we get vesicles up to about pH 9. This region has just spread out quite a lot. And if we have a 5-to-1 mixture of fatty acids to fatty alcohols, we're up to about pH 10. So we've got an extended region where we would expect to see vesicles now. And over here, the reason I'm showing these is this looks so messy, you wouldn't believe we could do any science if I only showed you this. If you take it at face value, it kind of says there's vesicles everywhere. And this, in fact, is the case. So these pictures are showing you 70 degrees centigrade, modern ocean salinity, and pH 12. And this is, these are cryo-EM pictures of vesicles with a bilayer membrane, and these are confocal microscopy, again, showing vesicles or protocells, if you'd like to call them that. I would like to call them that, but I appreciate it's a loaded term. But showing very clearly that you have vesicles with an aqueous lumen inside. So the idea that you can spontaneously form protocells from fatty acids formed under hydrothermal conditions will form relatively robust structures under those conditions is quite pleasing. It's also quite interesting. We get all kinds of wild and wacky structures depending on the salinity, for example. Sometimes we get stringy things. And so at the top here, these are microfossils that have been discovered in ancient rocks from the Archean and the Hadean, late Hadean. And down here are the structures of vesicles that we've got. And we sometimes see long kind of spindly chains of vesicles when we look more closely. So I'm not really suggesting that some of the cell-like things that have been found in ancient rocks are in fact not cells, but just fatty acid vesicles. But we shouldn't rule out the possibility that there's a kind of continuity between the structures that are formed just by mixtures of fatty acids and water, salt water, and the kind of structures that have been interpreted as cells by people studying Archean rocks. So there's some continuity there and some difficulty in distinguishing the morphology. So in terms of can we make iron sulfide nanoparticles? That's what we were expecting to make. What we actually saw was quite a surprise. What we saw was the shoulder on UV-vis spectroscopy, which is the characteristic diagnostic shoulder of 4Fe, 4S clusters. It was frankly the last thing I expected to see. We looked at how that shoulder changed under various different conditions. So we looked to see different cysteine concentrations, different ferrous iron, ferric iron, sulfide, and so on. And we were getting different concentrations of 4Fe, 4S clusters under those various different conditions. And we could back that up with Mossbauer spectroscopy. So this is a mixture of 4Fe, 4S, and 2Fe, 2S, and mononuclear iron clusters. And they also vary with the cysteine concentration, 3.5, 5, and 10. So quite a lot of variation over a short range. And we needed lower concentrations to form the 4Fe, 4S clusters. Also there, from cyclic voltammetry, we can see that the midpoint redox potential is around about minus 450 millivolts, which is very close to the ferredoxin midpoint redox potential and in the right range to be reducing CO2. Okay, so that's all I'm going to say on CO2 fixation. For metabolism, let's pretend it's true. let's imagine that we do have some kind of protocell. This is shown here. This is a mathematical model shown, so it's shown kind of wedged in a wall, which is not what we're thinking of. What we're trying to have is 50% exposed to CO2 and protons and 50% to alkaline solutions, so that we can have a continuous flux of protons driving this kind of branching metabolism. So C2 would be an activated acetate. So something like thioacetate, an equivalent to acetyl-CoA in modern biochemistry. And that is a Krebs cycle intermediate, giving rise to sugars, to some kind of an energy currency, to fatty acids, and to two different types of amino acid, one of which is something like cysteine, which could bind to these iron sulfur clusters. So we depicted them here as nanoparticles in the membrane. Now I would say they're iron sulfur clusters, but cysteine, again, would feed back, form more iron sulfur clusters and increase flux through the system. So we have a positive feedback loop that if you can make amino acids like cysteine, they will interact with iron sulfide and sodium, sorry, iron chloride and sodium sulfide to form 4Fe4S clusters, which can drive CO2 fixation and increase flux through this network. And if that happens, we can perhaps get as far as nucleotides. And there are a bunch of cofactors that are nucleotide cofactors. So the ones that are most relevant here would be folates and pterins, which are used by archaea and bacteria for CO2 fixation. So there's another positive feedback loop here. They're derived from GTP. But there's also, you can think of NADH, you can think of ATP itself. These are all nucleotide cofactors driving chemistry. So in principle, we could have those feeding back on all of these paths. And because this is just an in silico mathematical model, we did that. And we could have them feeding back on their own synthesis. So you need ATP to make adenosine these days in biochemistry. It could be done with a different phosphorylating agent, but nonetheless, there's a positive feedback loop here, kind of an autocatalytic cycle. So we were able to show that there are conditions where they will increase the rate of protocell growth. So the rate at which these kind of double their content and their membrane surface area. And we just assume then when you double the surface area, they spontaneously divide in two. All of these pathways really didn't help. You tend to funnel flux down one pathway at the expense of the other pathways. And interestingly, that collapses growth. But it helps if we have this pathway feeding back on CO2 fixation and this pathway feeding back on nucleotides. So if you make more nucleotides by increasing that, and they have a catalytic effect back on CO2 fixation, then over here, we're looking at protocell growth, and this top right-hand corner is the fastest growth rate. So the axes here, autocatalysis, so that's this step here. So if we've got a fast rate of autocatalysis of nucleotide formation, and we've got a fast rate of catalysis of CO2 fixation, then those two things together will drive a fast rate of protocell growth. What we kind of were hoping for was that under some set of conditions, we would also be able to accumulate nucleotides. And we thought if we increase the nucleotide concentration, maybe perhaps more likely to polymerize. That's probably a bit silly, but in any case, it doesn't work, because if they're catalysts, they drive cell growth and cell division, and then half their concentration every cell division. And so it only really works down here where they're very bad at CO2 fixation, but very good at autocatalysis. And then you don't have much, we know from this that you don't have much protocell growth. So it provides some interesting constraints on how the system as a whole can work. And it suggests, weirdly, that the overall thermodynamics of this system across a network of metabolism is already about as balanced as it gets. And if you imbalance it by pushing more flux down one pathway, it tends to worsen cell growth rather than improve it, which was, again, an unexpected finding. So this is the universal core of biochemistry as we know it. So this kind of chemistry that I was just showing you in theory, this is what it actually looks like, starting with CO2. And I just really want to register here hydrogen, hydrogen, hydrogen in brown, almost everywhere you look, and CO2 in green. This is, the core conserved biochemistry of cells is the chemistry of hydrogen and carbon dioxide. And then to get to amino acids, NH3, and the amino acids are coming from Krebs cycle intermediates, as I said before. In the red boxes here, these are the precursors for nucleotide synthesis following life's chemistry, life's biochemistry as a guide. The blue arrows here are things that have been done in the lab, either by us or other groups. There's several groups working on kind of metabolism first, this spontaneous metabolism. Another thing to say about this is that this chemistry is conserved across all of life, but the enzymes that are catalyzing it are not always conserved in the bacteria and the archaea. They can be quite different and not deeply conserved at all. And what that seems to suggest is that the chemistry of life is more ancient than the genes that are supposed to code for that chemistry. That would have been a really radical statement 10 years ago. I don't think it's particularly radical anymore. So that's a sign of how this field has moved. It's now looking increasingly likely, because so much of this chemistry is being done in the lab, albeit under different types of conditions. It's looking increasingly likely that the core of biochemistry really is a thermodynamically favored kind of topological chemical network. I just want to give you a couple of examples of some of our own work on this. So we wanted to see if we could make aspartate and started very simply with oxaloacetate. And in the presence of pyridoxamine, which is an organic cofactor, so you could say we're cheating, we're also trying to make pyridoxamine. Interestingly, this is aspartate here with pyridoxamine, but no metals, we don't see anything at all. But if we've got copper, this is a different types of metal catalyst here. Copper, we get quite a lot of aspartate. So copper plus pyridoxamine, we get quite a good yield. Ferric iron plus pyridoxamine, we get a reasonable yield. But all these other metals, including things like vanadate, don't really seem to work very well at all. We get much higher yields of alanine. So to get alanine, the oxaloacetate that we're starting with, which is not very stable, has to decarboxylate, so it loses a CO2, and then it reacts with pyridoxamine, according to this step here, to form alanines. So that's happening much more readily than aspartate, but it's interesting. So we're getting both, and only under certain conditions do we really get aspartate. But this, I think, was the first time anybody had succeeded in synthesizing aspartate from oxaloacetate. So nobody's really yet succeeded in synthesizing oxaloacetate directly. So there's plenty of challenges remaining. This is synthesis of uracil. The reason we wanted to make aspartate is because it's one of the substrates required for uracil synthesis. So ammonia carbonate to form carbamoyl phosphate and aspartate to form carbamoyl aspartate, then into dihydroorotate, orotate, and uracil. And this is a one-pot synthesis, starting from carbamoyl aspartate. We've now done it also from ammonia and carbonate, under various different conditions, in this case, switching pH as we went through. And this is orotate yields, and this is uracil. So that's a one-pot synthesis. These are the bases. These are not the nucleotides, but nonetheless, this entire pathway can be done as a one-pot synthesis following the biochemical pathway. And the bit in the red box here, we tried to optimize that, and that's what you can see here under the optimal conditions. So these were the standard conditions at the beginning, where in this red box we were getting orotate is the black, uracil is the orange, and under the optimized conditions we were getting around about 8 times the yield of uracil. And those optimized conditions turned out to be pH 8.5, 90 degrees centigrade, 1 molar sodium chloride. And so again, not unlike alkaline hydrothermal conditions. So again, all I'm really trying to say here is quite long metabolic pathways involving a number of steps, 4, 5, 6 steps, can happen spontaneously. And this is one PhD student over a couple of years doing this from scratch. So it can be done much better, no doubt. But other people have also published something very similar. In fact, they beat us to it, which is why we've not published this yet. Other work from our lab, we wondered if we could make acetyl phosphate as an alternative energy currency to ATP. So this is just a 2-carbon. So I mentioned the 2-carbon. So acetyl phosphate is basically acetate with a phosphate on it. These are different pH. The blue is at room temperature. The red is at, I think, 50 degrees. And this is the yield of acetyl phosphate in the presence of magnesium ions and calcium ions at the different temperatures. So better at pH 7 and this is magnesium and calcium ions, and we're getting reasonably high. Time is in minutes here, so within a couple of hours, and really within a very short period of time, we're getting you know, in a matter of minutes, we're getting more or less the maximal yield. And over here, this is phosphorylation of ribose at 25 degrees centigrade and 50 degrees centigrade, and down here is phosphorylation of adenine to AMP, of adenosine to AMP. So again, these are all biochemically conserved reactions that we're showing here. And again, it's happening in the presence of metal ions, but without any other catalysts around. And even more excitingly, this was actually repeating some work from Japan in the 1990s, but under a wider range of conditions. And it shows that acetyl phosphate will phosphorylate ADP to ATP. So the same reaction that the ATP synthase is catalyzing. This is with ferric iron alone in water as a catalyst, and we're getting a 15 to 20% yield. It works better at mildly acidic pH, so it's pH 5.5 to 6. Doesn't work at pH 9. This is the temperature. So it goes very fast at higher temperatures, but it tends to hydrolyze afterwards. At lower temperatures, you retain that yield. It worked with acetyl phosphate. It didn't work with other phosphorylating agents. It worked with ferric iron, but it didn't work with other metal catalysts. And interestingly, at the right-hand side, it worked for ATP. So we phosphorylate ADP to ATP, but we were unable to phosphorylate CDP to CTP or GDP to GTP or UDP to UTP. So if you wonder why ATP is the universal energy currency, at least part of the answer is, well, you can form it in water with ferric iron as a catalyst and from acetyl phosphate. So again, it's kind of, this is what's happening in bacteria as a form of substrate level phosphorylation, going from acetyl-CoA to ATP by this intermediary of acetyl phosphate. So this is, again, chemistry that we know about from biochemistry, and which we can do in water under remarkably simple conditions. So I promised you I'd talk a little bit about chirality. And this, well, I gave a talk to the Guy Foundation. Some of you may have been there some months ago, earlier this year. And I talked then about anesthetics and complex 1, so of the respiratory chain in mitochondria, and the possibility that anesthetics might disrupt chiral-induced spin selectivity. So I'm not going to go over that ground again very quickly, except to remind those who are here that we have electron transfer in complex 1 through this chain of 4Fe, 4S clusters, most of them. And that seems to spin polarized electrons as they're traversing across complex 1, and anesthetics seem to disrupt that. So it's by no means proved that that's how anesthetics work, but it is an exciting possibility that they may work this way, and it brings in a quantum biology element to consciousness as well. So this is again just showing, so this is slightly closer, these iron sulfur clusters. I've touched on this before. The electrons traversed by quantum tunneling from one to the next. The distances appear to be carefully selected. If they get more than around about 15, 16, I think the limit is close to 18 angstroms, then you don't get any electron transfer by quantum tunneling. So this is the structure of the 4Fe, 4S clusters in complex 1. Now, complex 1, interestingly, is known to be, and this was discovered by Manuela Pereira among others, but it's effectively a missing link, which is to say the energy converting hydrogenase that I was talking about. So I'm trying to just drag this back over here somewhere. Can't do it. Okay. So this is the structure of complex 1, and in red is the ECH, and then these other colors down here inside the membrane. The membrane is the parallel lines here. These are sodium proton antiporters. So complex 1 is a structure, and if you then look at ECH, the red one here as well, the membrane component of that is also a sodium proton antiporter. So the membrane component here is basically 4 sodium proton antiporters that have been split somewhat with a little bit extra inserted in the middle. So a very interesting and peculiar structure. And there's a paper just a month or two ago from Tonio Enriquez in Madrid showing that it still works as a sodium proton antiporter. So the proton motive force is partially converted into a sodium motive force in complex 1. So this is the membrane protein I was telling you about with the 4Fe, 4S clusters that are driving CO2 fixation in archaea. Here it is, it's sitting right at the heart of our own complex 1 that's in our own, this is actually the bacterial version, but we have exactly the same protein in our own mitochondria. So if there is quantum tunneling and electron spin polarization going on in complex 1 now, then similar things should have been happening here, although there are fewer iron sulfur clusters in that. Well, what grabbed my attention was a paper from a year or so ago from Furkan Öztürk on the possibility that chirality was introduced into biochemistry by magnetic surfaces. And these are pictures taken from his paper. This is with John Sutherland, among other people, who is from, let's say, the other camp insofar as they are interested in an RNA world, and they are starting with cyanide and cyanacetylene and so on. One of the starting points that they had is RAO, which is ribose aminooxazoline, and what they're showing. So to get to ribose, if this is forming the D-ribonucleotide on a mineral surface, so the mineral magnetic surfaces, if they are interacting with different enantiomers, one enantiomer will tend to be drawn to that surface, and there's plenty of people in this audience who will understand this much better than I do. All I can really say here is that one of the two will tend to bind to that surface, and the other one will tend to be repelled from that surface. So these are the enantiomers. And they also show that it can, in fact, induce that if only one enantiomer is present, then it can, in fact, magnetize that surface locally. So you then have a positive feedback loop going on. So they're starting out to form this RAO, and its binding is slight enrichment in one enantiomer. So the D form is bindings to this surface. It can be recrystallizing onto that surface, which is going to induce some level of magnetization, which is going to lead to higher selectivity. And so you've got this kind of feedback loop, which is going to lead eventually to more or less pure enantiomer selection of the D form, which goes on to form D-tRNA. And what they had shown, and this is an idea that goes back in the literature some way, but if you're making D-ribonucleotides, you will then make D-transfer RNA, and D-transfer RNA will tend to select for the opposing amino acid. So you would select for the L enantiomer of amino acids. And so you've got a system which can then feed back into metabolism, which is capable of effectively forming the stereospecificity that we know in biology. So this is exciting work, I think. The problem that I have with it is that I don't believe that we're starting with the RAO here. And so the question is, well, can something similar be happening following protometabolism as I would like to imagine it, rather than starting with cyanide? So here's summarizing that. So RAO is ribose aminooxazoline. It's a synthetic intermediate to RNA starting from the condensation of cyanamide and glycolaldehyde. And it's very different to the biochemical pathway. So I immediately wonder, could CO2 fixation on magnetic surfaces, and that would include magnetite and greigite that they were using, also it can include 4Fe, 4S clusters when they're in the +2 state. They are magnetic in the +2 states, these clusters. So could you select for chiral intermediates in metabolism on magnetic 4Fe, 4S clusters is the question. And the answer that I've come to over the last couple of weeks since giving this title is no, I don't think so. I'm afraid that chirality is going to be lost in the Krebs cycle again, because most of the intermediates of the Krebs cycle are not chiral. And it would lead to a problem in amino acid synthesis that some are coming from chiral intermediates, some not. It becomes very messy, and that's not really the answer. But a better question is there a step in gluconeogenesis, which is involving iron sulfur clusters and so on, where the same chirality could be imposed? So specifically in gluconeogenesis to form D-phosphoglycerate, for example. And the answer there is, well, yes, potentially. So this is not my work. This is coming from Joris Zimmerman and Joseph Moran, and this is taken from his PhD. So this is available online. And what you're seeing here, these are Krebs cycle intermediates, and this is coming into gluconeogenesis. It could go either way. But they have done these reactions going from phosphoenol pyruvate through to phosphoglycerate here. So I'm just trying to move this thing out of the way again. And they've shown in this diagram where iron is involved in, this is all prebiotic chemistry, this is not modern biochemistry, but these are pathways that have been done by the Moran group, who've been probably the most productive, prolific group of prebiotic chemistry using life's modern metabolism as a guide to it. So it's interesting here that fumarate is malate is chiral, but fumarate is not. The connotate is not, isocitrate is, citrate is, oxaloacetate is not, and here we're going from phosphoenol pyruvate, which is not, to phosphoglycerate, which is. So iron is involved in all of these switches from non-chiral to chiral, which is really interesting in this prebiotic chemistry, including, so this is the first step of gluconeogenesis here, and they have succeeded in doing this, albeit it's very difficult chemistry in the lab. And the next step from here, which is not shown, is going to glyceraldehyde phosphate, which, because this is the D-phosphoglycerate, goes to D-glyceraldehyde phosphate. If they've not looked at the stereochemistry, but if there were a magnetic effect here, that's the stereochemistry you would expect to see. And this is also work considering the aldolase. So there's all kinds of other chirality in for example, fructose bisphosphate. But this is starting from glyceraldehyde phosphate. This is the D form of glyceraldehyde phosphate, but you have an equilibrium with dihydro, see if I can remember, DHAP, so dihydro, oh God, acetone phosphate. So there's an equilibrium mix here, there's some messiness here, but effectively this chiral center is conserved here. There are other chiral centers are introduced, but none of them are important to the chirality of RNA. This is the center which is important to the chirality of RNA, and this is by the time we got to ribose. So the chirality of D-glyceraldehyde phosphate is conserved all the way through into ribose and would form D sugars, D-ribonucleic acids. So in principle, that one step is able to introduce chirality into all of biology. Nobody's shown that that happens, but it's possible that it could. Okay, that's all I have to say about chirality, but there's an interesting possible mechanism of magnetic surfaces and chiral-induced spin selectivity that can select between enantiomers and give you chirality in biology. It looks messier coming from the kind of metabolism that I've been talking about than it is from their chemistry, I have to say. But I don't like their chemistry, so I would rather find a different way of doing it if possible. Okay, information. I suspect people might be flagging. I'm going to be about another 5 or 10 minutes. I'm not keeping a close eye on the time. I should stop soon. If you can bear with me for 10 minutes, I'm going to do information. Origin of information in biology. It's hopefully sufficiently exciting as I'll keep your attention. So there are patterns. I mentioned there's patterns in the code. And the first base of the codon is linked with, it turns out, well, we've known for decades that it's linked with the biosynthetic precursor, but it was never really very clear what that meant. But if we look at it in terms of distance from CO2 fixation, if it's a single step reductive amination from a carboxylic acid, it's always a G at the first position of the codon. And if it's a few more steps, but usually involving ATP in one of those steps, then it's an A at the first position of the codon. And with much further away, it's a U at the first position of the codons, and this is a C, in blue is a C. So we have this rough relationship, between amino acids with a G at the first position of the codon, which are quite close in the number of steps to CO2 fixation, going to U, which is the most distant. This is cysteine, which we have reasons why we think that that's an outlier in this. We think it was introduced late into the code, but is nonetheless an early amino acid. And it may be that it forms a cyclic form, which is why it was introduced late into the code. So there's some suggestion, all I'm really trying to capture here is that there is some suggestion that one of the patterns in the code, which links with the biosynthetic precursor, in fact links much more cleanly with the distance from CO2 fixation. And so there's some link with autotrophic protometabolism and the genetic code. Difficult to say exactly how that link works, but there's definitely an association to be seen there. So if we stick with that link and we say there's a G at the first position or an A at the first position, so these are the ones closest to CO2 fixation, and these are the ones further away, these are the pyrimidines are further away from CO2 fixation. And then we're looking at the second position of the anticodon, grouping them according to distance from CO2 fixation. We're seeing an association between the hydrophobicity of the amino acid and the hydrophobicity of the base that codes for it. So in this case, valine, for example, color coding has changed, which is a little bit irritating, but the gray is A at the first, at the second position of the anticodon, and orange is a G, and so on. So A, adenosine, is the most hydrophobic of the bases. And it codes for valine, which is the most hydrophobic amino acid. And this is 43 different scales of hydrophobicity that we've taken into consideration here. And you can see there's a fair amount of variation between those scales. Some of them are solubility in organic solvents, and some of them are associations within proteins. And so there's all kinds of different ways of measuring hydrophobicity, and they don't agree with each other very much. And there are more sophisticated ways of talking about it, partition energy, this kind of thing. So this is probably not particularly granular, but what we are seeing here is the more hydrophobic amino acid interacts with the more hydrophobic base. And at the other end, uracil, which is the most hydrophilic base, interacts with the most hydrophilic amino acid, which is aspartate. These are the group which are the closest to CO2 fixation. We see a strong association as well with A at the first position, again, the most hydrophobic amino acid, the most hydrophobic base, most hydrophilic, and so on. That pattern seems to disappear further away from CO2 fixation. So it's really very clear in early metabolism. And again, we don't have very clear explanations for that. It's just that it's a very clear pattern. And at the third position of the codon, this is an inverted codon table. So just to show you what we're seeing here, so glutamate and aspartate, they both have a G at the first position of the codon. They both have an A at the second position of the codon, but glutamate has a purine at the third position and aspartate has a pyrimidine at the third position. So that's of the codon, it's inverted for the anticodon, and effectively purines are larger than pyrimidines. They're two ring structures rather than one ring structure. And what we're seeing here is that we can predict which of these it's going to be based on size alone. So the larger amino acid goes with the smaller base, and the smaller amino acid goes with the larger base, and so on. So there's a rather, not only is it hydrophobicity, but it's size as well. So there are stereochemical interactions which are associated with the pattern of CO2 fixation, with metabolism and CO2 fixation. And the earlier the closer to CO2 fixation, the stronger those patterns are. So again, there's something going on. Well, this is just looking at the code and saying, oh, there's patterns here and we're interpreting them this way. Is there any reality to it? So this is molecular dynamic simulations. And what we're seeing here, for example, is we have proline and we have each of the four bases, and we look to see, this is AMP, GMP, CMP, UMP. This is the proximity and this is the time spent within that distance. So within 2 angstroms of proline, GMP spends quite a lot of time, whereas the rest spend much less time. In other words, the molecular dynamic simulations are saying that GMP likes proline. It likes to spend time next to it. Well, GMP is in fact the middle base of the anticodon that codes for proline. So we got that right. I'm showing another example here, which is much more equivocal. In fact, we get this right for arginine as well, but you can see there's much more overlap here. So it's a little bit messy. And overall, these are our guesses, number 1, number 2, number 3, number 4, in terms of how much time spent there. And then in ring is the correct answer. And you can, and this is ranked in order of hydrophobicity. So these are the most hydrophobic over here. These are the most hydrophilic amino acids over here. And you can see we get 50% of them right. So we correctly predict on the basis of the molecular dynamics 50% of the mid-codon bases. But we do much better for hydrophilic amino acids than we do for hydrophobic amino acids, where we do pretty badly. So the molecular dynamic force fields, and we use 2 different force fields, do not predict hydrophobic interactions very well, but they do predict the hydrophilic ones well. And actually these interactions that are suggested from the code are found in silico, at least. We do better than 99% of random predictions. Then we've done some NMR, a limited amount of NMR. That we've done 5, I think, amino acids by NMR, and this is the binding constants. And so the lower it is, the tighter the binding. And again, we're seeing in the red boxes are the correct ones. These numbers are the position of the protons on the NMR on that molecule if there's more than one proton. And again, we're more or less getting it right for some of them, but not others. We get about 50% of them right. And again, we're doing worse with the hydrophobic interactions, in part because trying to dissolve a hydrophobic acid and a hydrophobic amino acid and a hydrophobic base is hard to dissolve in water, and we have to keep the conditions the same for all of them. And so we basically have a much weaker signal with the hydrophobic amino acids and the hydrophobic bases, and it's harder to interpret. Okay, last 2 slides. Let's pretend it's true. Let's pretend there are physical interactions between a random piece of RNA and the amino acids that then bind to it depending on their size and their hydrophobicity, and somehow, I'm not specifying how, somehow polymerize so that the sequence in the RNA is going to specify weakly a sequence of amino acids in a peptide. And so this is an in silico model from Raquel Nunez Palmera. And so we're doing CO2 fixation to make purines and pyrimidines and hydrophobic and hydrophilic amino acids. We're polymerizing the purines and the pyrimidines, free polymerization, so not templated at all. Then we're doing some copying. So a purine binds to a pyrimidine and so on. Then we're doing translation. So we're just assuming direct templating here that the hydrophilic amino acid binds to a hydrophilic base, a hydrophobic amino acid to a hydrophobic base. Then we assume some decay, that things fall off the end, and then we go through that cycle again. But we assume some positive feedbacks that if we are beginning to select for better CO2 fixation, better copying and so on, so we're assuming then catalysis, that some of these peptides are better than others at catalyzing CO2 fixation, and we assume that some of those could be better than others at catalyzing copying or templating, so these processes of copying and translation going on over here. And then we can model that to see what the outcomes are. And well, I'm not going to spend long on this, but if this is the simplest version where we're looking for hydrophobic amino acids and hydrophobic, so a hydrophobic peptide will go to the membrane, for example, and catalyze CO2 fixation. A hydrophilic peptide would stay in the cytosol, bind to a metal ion, and possibly catalyze copying. So an RNA polymerase is a magnesium-dependent protein. It's always aspartate that binds to the magnesium ions. And so we're looking for a breaking of symmetry here, where we're forming, if it were just random, we would be forming a kind of cloud in the middle, a little bit like this. But what we're actually forming, C, for example, here, is a P of 0.005. So this is the free polymerization rate. So that's this one. So we're seeing different rates of free polymerization and very fast rates. If it's just free polymerization, it's noise. You're producing random nucleotides all the time. And so you're constantly making it noisier and you get a lot of random noise. If you have a lower free copying rate, then what you end up with is less noise and the possibility of selecting optimal sequences, which we're seeing here as these kind of clouds which are going towards the axes. Now, that's a very symmetrical and somewhat stupid case. The question was, could we alter, could we specify some particular optimal sequence for CO2 fixation or for RNA copying? And we can evolve towards those. It becomes quite messy, but we can evolve towards the optimal sequence spaces in some cases, but not others. What we actually found is that you must do CO2 fixation first. You must evolve more easily a sequence that will drive CO2 fixation and growth than anything that will do copying. Because to copy, you have to double everything all the time, and that just means you need more materials. And if you don't have the more materials because you're not doing CO2 fixation, you can't grow. So it's an interesting order. It's in the title: first growth, then information, the path to genetic heredity in protocells. That's not out yet either. So that's all I really wanted to say. That's perhaps I've said enough, so I'm not going to dwell on the conclusions. But all I'm really saying is that we have electrical membranes, proton-motive force across membranes, driving a spontaneous protometabolism, giving rise to genes all in one place. The people who've done this work, I'm very grateful to them for sticking with me and for asking really difficult, exciting, but dangerous questions in biology. And some of them have been with me for some years now. Some of them, a couple of those have left my group, but and also to the funders, and thank you. I'm very happy to take questions.